including the NSF Career Award, Sloan Research Fellowship, and Tufts Emerging Leader Award. Dr. Jessica, thanks for the seminar, and the floor is on yours.
Thank you, Tim, for inviting me over. It's my great pleasure to present my work here. And also, I want to say a special thanks to my former student, Keshin, for joining from Microsoft. She's now working there, and she contributes to the work I will talk about today. So I pick a pretty big title, I would say, simulator, because I think it is something that we can potentially contribute to the biomedical data science community and try to bridge the abstract thinking in statistics and the real data analysis. So I think simulator is a way to make our model more concrete and transparent to users who may or may not know so much statistics. So what I think, these are the reasons why I think that simulators are useful and why what are the requirements to make them useful. The first goal is, for sure, I think it's the most easy reason people can think of, is to benchmark computational methods. And that's why many simulators are used in method development. But there's always a criticism from scientists, that is, simulation is never the truth. Then why would they trust them? However, my argument is that if the simulated data mimic real data to a certain degree, then we should conclude that the simulated model is useful. And there's a famous quote from George Box: "All models are wrong, but some are useful." That's the reason why we still use models, even though we know statistical models are not mechanistic models and they do not represent science. Second goal, I think, is to use the model. If it's useful, if it's reasonable, we can use it to help interpret real data. And this is what statistical inference is all about, right? We want to hopefully use the model parameters to inform some underlying ground truth behind our data. The third goal is to possibly generate some alternatives or variants of the real data. For example, power analysis is essentially doing this. We are generating different possibilities of the data and see how powerful our method can discover the signals. So therefore, the simulator model parameters should be modifiable to reflect hypothesis. So these are the three requirements I can think of to reflect, to answer, to achieve the three goals. So for this talk in particular, I will talk about our efforts in building a statistical model for single cell and spatial omics data. And the current version, we call it Essay Design 3. And I want to introduce it both as a simulator and as a model. So the first data or example we want to be able to mimic is the single cell RNA-seq data with discrete cell types. So this is one of the most famous data sets called PBMC data. It's from human blood. And we have very good knowledge about the cell types, which are labeled here. And the colors represent the cell types. And typically, for the data we get, we will perform some clustering analysis and then label each cluster as a cell type. Second example is that single-cell RNA-seq data may represent some continuous cell trajectory, such as cell differentiation. So in this case, we're not dividing cells into discrete cell types. Rather, we are ordering the cells in a continuous trajectory, and this trajectory is inferred from data. And we hope that this trajectory, which represents an order of the cells, reflects some dynamic process underlying. The third example is the more recent technique called spatial transcriptomics, which provides the two-dimensional cell locations in the tissue slice. So here, each spot represents actually multiple cells because this technology, 10x Visium, is not so high resolution. So every spot is much bigger than one cell. And you actually have the spot corresponding to multiple cells. And each slide here shows one gene. We're showing the expression pattern of the gene in the tissue slice. So for this type of data, you have some additional spatial information. But what I want to say here is that we can actually unify these data into the following format. So the data matrix is an n cell by n feature matrix. So the number of cells typically range from 10 to the third to 10 to the sixth. And the number of features depend on the technology. They could represent genes for RNA-seq or transcriptomic data, and the gene number can also depend on the technology, or they could represent some DNA regions if it's other types of omics. But this is the data we get out of the technology. Besides this data matrix, we typically have a cell state matrix. So this is my terminology, but I want to use it to unify discrete cell types, continuous cell pseudotime, or the cell spatial locations. I call all of them as my cell state matrix because I think these states determine the data, determine the omics features for each cell. And sometimes we may also have a design matrix. A design matrix is about the data collection. We may have data collected in different batches. So we have a batch character vector. And we also can have multiple conditions or more than one condition to study. And typically the between condition differences for each feature is of our interest, while the batch effects are not of our interest, and it's a confounder we want to remove. Then why do we need a statistical model for such type of data? Just for your information, as these technologies are so popular, there are numerous algorithms for analyzing them, and they are not necessarily using statistical models. I think a statistical model will be helpful because if the model parameters are interpretable or reflecting certain scientific hypotheses, then it's useful. And we can use the model parameter inference to tell us some biology. For example, what is expected expression of a feature like a gene given a cell covariance, say given a cell type, what is the true expression level of a gene? And statistical inference can give us some uncertainty quantification for the parameter estimates. So we are more confident or less confident about our conclusions. The second purpose, as I mentioned already, is to generate some alternative to real data, for example, power analysis. But to do this, we should be able to sample from the model, right? So the model should enable some easy sampling, fast sampling. Furthermore, I want to say that model diagnostics is very important, but it's sometimes ignored in our statistical method development. Maybe we say, oh, we propose this model, we do some inference, but how reasonable the model is, or how well does it fit to the data? That's often not discussed. But I want to emphasize that here, I think model diagnostics is very important. So I have the following considerations for modeling such type of data. First of all, I consider the cells as samples from a cell population. So the cells are considered to be random observations. I think this is consistent with the technology or the experiment because given we draw blood cells, we measure them, but the cells we measure is actually a sample of all the cells. We cannot measure everything in a person. And we consider the genes or regions as features. So naturally, for this data matrix, I hope to have some m-dimensional distribution, the joint distribution of the m features. I want to learn that based on the n cells as samples. But for the cell states and the design, I want to be conditional on them because they are the things that we should account for. And given a certain cell state and given a design covariance, I want to know what is the corresponding m-dimensional distribution. So I'm interested in learning the conditional multivariate distribution. So along this line, my group has actually multi-year efforts for developing this model. The first work we published was with a former student, Vivian, in 2019, and we titled this work Assay Design. At that time, we just started, and I think our model assumption right now, if we look back, it's very naive. We assume the M genes as features to be independent. We just model the marginal distribution. And in the second version, which we published 2 years later, we made the model more realistic by considering the pairwise correlations among the features. So we are going from first order modeling to second order modeling. But in assay design 2, we only consider the cell state covariate to be discrete cell types. So we don't consider other scenarios like trajectory or the spatial location. Motivated by that, we have the current version assay design 3. It's more flexible. It can have beyond cell type covariates, including cell pseudotime along a trajectory or cell spatial locations. Furthermore, we go from modeling the counts, which I'll talk about. Basically, the data matrix, every feature in every cell is measured as a non-negative count. We go from counts to reads, because the reads are the raw data from sequencing experiments, right? So people just summarize reads into counts and start from there. But we think for some low-level tools, they start from reads. So therefore, it would be good we can simulate read. So that's the read simulator. And I want to thank all my students for leading these efforts, in particular Vivian for the first version, Tianyi for the second version, and Guangao for the read simulator. And as I will focus more on the third version, SDN3, so I will make an acknowledgement in the end. So for SDN3, which I will focus on today, was led by my current student Dongyuan Song and Qingyang Wang for their efforts in making this package in this current form. What we hope to achieve is to first do simulation on the left. So simulation is essentially sampling from our fitted model. Second use is to use the fitted model to get some interpretation. So let's first look at sampling, what we want to achieve. We hope to be able to generate multi-omics features, not just gene expression, but also the open DNA regions, proteins, DNA methylation. For the cell states, we hope to generate discrete cell types, continuous trajectories, and spatial data. For the design, we want to allow for biologically valuable covariates like gender, age, disease, health as conditions, or unwanted batch effects. On the right-hand side, we want to use our model, estimated model parameters to do the following. First, the parameters, we hope them to be useful. And for example, if the cell state is 2 cell types, then we hope to have this distribution of a gene's true expression levels in the 2 cell types. Then we can use this to say whether the gene is a truly differential expressed gene or not. We also want to fit a true expression trend of the gene along the trajectory. So it's like showing how the gene changes at its true value from the model parameter. We also want to learn a true 2D surface function for each gene in the spatial data, so we can use that to tell the gene's pattern. Besides that, a very important aspect in statistical modeling is model selection. If we want to use our model, if we trust the model to be reasonable, then we can use it to decide whether if I have 2 pseudotime or trajectory inference algorithms that give me different pseudotime values for the cells, I can input each set into the model and calculate the BIC to see which one is better. Though I can choose between 2 sets of pseudotime values. And I can choose between 2 sets of clustering results to see which one give me a better model fitting, and I choose that. Finally, this is the alternative generation part. I can modify my model parameters to make a gene to be non-differentially expressed or truly differentially expressed by changing the model parameters. And I can even, assuming that there's only 1 cell type, I can just fit a 1 cell type model to real data, just like I'm maximizing a likelihood under the null hypothesis. Then I can generate data by sampling from the null model, and I call that synthetic null data. So these are the things we want to achieve. And I will detail our model and how we can achieve this in the following slides. So just to quickly summarize, I use the following notations. Yij as the count for gene or feature J in cell I. And corresponding to the cell I have a cell state covariate vector denoted by xi, which is p-dimensional. I have a design covariate called zi, which we only consider 2 categorical covariates, bi for batch, ci for condition. What I'm interested in learning is cell I's m-dimensional distribution conditional on xi and zi. That's my goal. Then what's the SDN3 model? As I mentioned, this is like an outline of the graphical illustration I did in the previous slide. Y is a matrix n by m, cell by feature matrix. X is an n by p, cell by state covariant matrix. So these p covariates could be cell type, then p is just 1 categorical variable. It could be pseudotime values of the n cells in p trajectories, because the trajectory may not be just 1 branch, it can be multiple branches. So to be general, if there are p trajectories, then there will be p continuous variables, each one indicating 1 trajectory. And it could be 2-dimensional spatial coordinates, then it will be 2p equals 2. For Z, the design covariant matrix, I consider to be m by q. In particular, the current implementation has q being 2. So we have 1 batch covariate, one condition covariate. That's the data, or that's the formulation, the variables. So our idea is to first model each feature's marginal distribution, then connect them into a m-dimensional joint distribution. So for the marginal distribution, it is conditional on xi and zi. We consider multiple choices for the distribution depending on the data. For example, for some data that are counts, then this Fj should be a count distribution like negative binomial distribution, and we may add zero inflation component to it depending on the data type. Or for some other type, Fj may be reasonably assumed as a Gaussian distribution. So it depends on the data and depending on the technology, I should say. And given xi and zi, and Fj at the most have three parameters: mu ij for the location parameter, sigma ij for the scale parameter, and pij for the zero inflation parameter. So that's the most complicated model we consider. And for the mean or location parameter, we consider it the mu ij subject to some link function depending on Fj. We consider the transform, the link predictor, linear predictor to be depending on a common intercept, a batch intercept. This is the intercept contributed by batch. We just model batch effect to be intercept, and there's a cell type intercept, and then there is I shouldn't say cell type, and sorry, condition, by that. Condition intercept, and this is a given the condition interacting with the condition, there is an effect of the cell state covariate. So basically the design covariates BICI entered in the two intercepts, and the condition effect also enters in this effect of XI, state covariate. So in the most complicated form, sigma ij scale parameters subject to log link function, Pij probability, zero proportion probability, zero inflation probability subject to log link function. They also can be modeled in a similar way, but I have to say that we don't necessarily use the most complicated model for certain data. It's just like we're writing out the most complicated form here. And to fit this model, we use this method, or we use the model called generalized additive model for location, scale, and shape, GAM-LLS. So that can fit this model. So we do that for each feature J. Once we fit the model, we denote the fitted marginal distribution for feature J as Fj hat. All right, that's the marginal modeling. So Fj, the xi, xj, zj, and the three things. So just to be clear, we need those nonlinear functions or non-parametric modeling for this f, g, and h if we have xi as pseudo time or if it's spatial location, because we don't want to make it assumed to be linear. But if xi is just cell type, then each one is actually just additive. It's just an intercept. All right, so these are the distributions we currently consider in our package. Like I mentioned, Gaussian, negative binomial, zero inflated negative binomial. We also have Poisson and zero inflated Poisson because for some data, it may be just Poisson is enough. We don't need over dispersion. And Bernoulli is also included for modeling binary data. For example, methylation data can be considered binary. And I already said this, for the cell state covariate xi, it could be discrete cell types, one lineage, then it's one-dimensional, continuous, right, from 0 to positive infinity. Or if it's p lineages, it's p-dimensional. For spatial location, it's two-dimensional. Then for the joint modeling part, what we use is the technique called copula to link m marginal distributions into a joint distribution. And the copula we use in particular is the Gaussian copula. The idea is that we first use each feature J's corresponding CDF. So right now we are using the fitted CDF because we are using a two-step fitting procedure. First to fit marginal distribution, then try to find parameter estimate for the Gaussian copula. But in the modeling, to write down the model, we just assume that there is a CDF for each feature. We use the corresponding CDF for each feature to transform the data into a CDF value between 0 and 1. So each one becomes a uniform random variable between 0 and 1. That's just the property of CDF transformation. And with that, the copula function is just trying to link m uniform 0 to 1 random variables into a joint distribution, into a joint CDF. And by Gaussian copula, what it happens is that with m uniform 0 to 1 random variables, it applies an inverse of the standard Gaussian CDF function to each random variable to change it into a standard Gaussian random variable. Then to fit the joint distribution of n standard Gaussian random variables, we just need to estimate an m by m correlation matrix because each variable has mean 0 and standard deviation 1, variance 1. Then we are left with estimating this correlation matrix. But there might be a dimensionality problem. That is, what if this m is very big, right? If this is very big, then we can have this problem that if we just estimate this correlation matrix by using a sample correlation matrix, we may not have the invertibility, then that could be a problem. So therefore, to estimate this correlation matrix, we use this technique called Vine copula, which is to help regularize Gaussian copula. And the idea is that you constrain the correlation matrix or the pairwise correlations of the m features in the matrix to satisfy this Vine structure. What it does is that you have the first layer just as the pairwise correlation of the most highly correlated variable feature pairs. Given the first layer, you want to conditional on that and get the conditional pairwise correlations for the strongest pairs, and then you go on. So this can ensure that the resulting matrix, correlation matrix, estimated correlation matrix is invertible. And to estimate, this is just what I said. So basically, that was the model. And to estimate the parameters, I think I already said some of them in the introduction. That is, we do the plugin. We first estimate the m marginal CDFs. But to apply the Gaussian copula, there's one thing we must consider. That is, if the marginal distributions are count distributions that are discrete, how can we make the variable turn it into a continuous Gaussian random variable? So there's something gap here, right? Because if you apply the CDF function, fitted CDF function to each count random variable, the resulting variable is still discrete. You cannot make it continuous uniform 0 to 1. So to make it uniform 0 to 1 continuous, we use this kind of random interpolation technique called distributional transform. We just randomly sample Vij from uniform 0 to 1 as the weight. Then we add up the weighted sum of the consecutive point masses at the two values. That's just the thing we did. All right. So suppose we fit this model, right? Gaussian copula for the correlation structure, marginally, we have each feature with its marginal distribution. Then we can do sampling. So when we do sampling, essentially, what we do is that we will first sample from the m-dimensional copula. Say, if I want to sample m-prime synthetic cells with the same m features as in my original data used for model fitting, then what I need is I also need to specify the m-prime synthetic cells state covariates, p-dimensional state covariates, and the q-dimensional design covariates if I use them. So I need to specify them because my model is conditional on them. So then putting them in the condition, I use the fitted copula to draw an m-dimensional vector. Each entry here is uniform 0 to 1. Then what I do is that I would use also my fitted parameters here, the location, scale, and zero inflation probability to actually get a marginal CDF. Okay, that's from the GAM LLS for the M features. Finally, putting things together, I will do an inverse of the fitted marginal CDF. I will apply that to each sampled U random variable, UI prime something, UI prime J, then I will get the YI prime J. So that's the synthetic data I get for the M prime cells. So M prime synthetic cells. So that's the most general framework. Here, I will illustrate the idea using our model for one cell type, two genes for illustration. So we assume this is like single cell data where single cell RNA-seq data where marginally every gene can be reasonably assumed to follow a negative binomial MB distribution. So in our marginal model fitting, we fit an MB distribution for each gene. Then second, we use those fitted CDF to transform each gene to CDF values. Further, we use the inverse Gaussian CDF to transform each gene to Gaussian values. And then we fit a multivariate Gaussian distribution, where here's two-dimensional Gaussian distribution. Then we can, from here on, we can do sampling. We can first sample M prime synthetic cells from the Gaussian distribution, but each gene is on the Gaussian scale. Then we apply the CDF of standard Gaussian to change it to CDF values. Finally, here the fitted marginal CDF inverse will be used to transform CDF values back to the count values. That's our synthetic data. So essentially, given this is one cell type, we are conditional on this cell type, and we don't consider design covariates just for simplicity. But basically, synthetic cells are just a sample from the fitted distribution. And the fitted distribution here is basically a two-dimensional negative binomial distribution with the correlation structure specified by Gaussian copula. And we only consider second-order interactions of the features. So just in our SCD-3 paper, we also have other options, right? Like I mentioned, we can generate cells in a continuous trajectory. And here we're trying to show that compared to an existing single cell deep learning model for generation using the GAN idea, we can actually do even better for mimicking the real cells. In particular, this index called mean local inverse Simpson's index is a way to measure the similarities of two groups of cells. So what it does is that in some low dimensional embedding space, you pull the cells together and you measure for every cell in its local neighborhood, what is the composition of the real cells and the synthetic cells. If the proportions of the two groups of cells is half and half, this index will be 2. That's the ideal case. If the neighborhood is dominated by its own group, then the index is 1. So this index varies from 1 to 2, with 2 being most similar. So you see that we're having an even higher index. And also, our model used the trajectory or the pseudo-time values of cells in the trajectory as a covariate. So we can still have the covariate output, like the true pseudo-time values for synthetic cells, while GAN doesn't keep this information. And maybe it's due to the implementation issue. This package runs very slowly. So we even have this advantage in computational time. So I will call this simulation as a way to check the model, because we hope that the simulated data will look like real data. This is spatial transcriptomics. The top row is the real data. Second row is our simulated data. And here we're just calculating a very simple Pearson correlation because we fixed the same spots. The spatial locations are 1-to-1 match. We didn't sample spatial locations. We just generate gene expression on the same locations. So therefore, there's a 1-to-1 map between the spatial locations, and we just compute the Pearson correlation for this gene across all the spots, and we found that the correlation is pretty high. And I forgot to say, for the function of the gene, the mean expression of the gene in the 2D space, we use Gaussian process. Yeah, that's just a special case for our model I didn't mention. CytSeq is another technology that allows you to measure RNA and their accompanying protein expression on the cell surface. So for these 3 immune markers, we can have both protein and RNA.
So what we try to show here is that besides mimicking just RNA protein separately, we also capture the relationship between the protein RNA. So it's like pairwise, we still have the similar protein RNA relationship in our simulated data. And this is like the trajectory example. So this is using our parameter estimates, the one we stated. And for this trajectory scenario, the effect of the cell state covariate, which is pseudotime on gene expression, is actually captured by a spline function. And we can actually see that a spline function provides a way for us to interpret the data, so which is another output from the model. So beyond the simulator. And this is how we fit it using the Gaussian process for each gene in the spatial transcriptomics data. So it's like a smoothing of the real data. So we can see some hotspot here. Finally, the Gaussian copula and its constraint version Vine copula can give us some useful information about pairwise gene correlations. So we have 4 cell types, and we can look at their Gaussian copula. This is just the sample correlation matrix we use to estimate the correlation from the data. And this is the Vine, corresponding Vine for the constraint, for the regularization. So what we want to see here is, if you look at the 2 T cell subtypes, their correlation structure is actually more similar to each other than to the other more different cell types. So we think this within cell type gene correlation is something very interesting, because obviously genes are not independent within a cell type. But what does this within cell type correlation mean? I think this is still a very open question in the field, because when people talk about gene correlation, sometimes they don't really differentiate, saying, okay, I'm talking about between cell type correlation across all cells of all types, or I'm specific talking about within cell type gene correlation. And we know that they're different because of Simpson's paradox. And I think biological interpretation should also be different. Finally, this is the model selection usage of our model. So we consider 3 pseudotime inference methods: T-scan, monocle 3, slingshot. They were all proposed to order cells and assign pseudotime values to the cells. So we calculate the BIC by inputting each method's pseudotime values as the cell state covariate into our model. So we can check the model fitting. Just as a reference, so basically the blue dots are from our simulated data where we specify ground truth pseudotime, so it's ideal. And we can see that the more noise we add to our ground truth pseudotime, the worse the BIC. So here we just want to show there's some monotone relationship between BIC and the R squared. So with our simulated ground truth. And for the 3 dots here corresponding to the 3 values, we don't have ground truth. So we can compare their BIC values in an unsupervised way. And we actually see that among the 3, slingshot seems to be doing a better job. And if you look at the pseudotime values, which are plotted as colors in this 2-dimensional UMAP embedding of single cells. So UMAP is just a technique for showing single cells in 2D. We can see that roughly slingshot seems to have a more smooth transition across the cells. Maybe that explains why we give us the better model fitting. Okay, for alternative generation, which I will talk about for the remaining of my talk. So this is something I think it could be interesting and useful. That is, if we assume our one hypothetical cell type model, that is the multivariate negative binomial model with Gaussian copula for the correlation structure. What we can achieve is by preserving every gene's mean statistic, every gene's variance statistic across all cells, and gene correlations across all cells. These are supposed to be preserved by our parameter estimation. But since our model is assumed to be homogeneous, and the homogeneity comes from 2 layers. One is that every gene is a unimodal negative binomial distribution. Second, the Gaussian copula is by nature homogeneous, right? We know Gaussian has this tendency to have density in the center, and the density decays as we get away from the center. So therefore, our simulated cells from this model will be like a homogeneous cloud of cells. In contrast to, we see this gap in the real two cell types. What is the use for this? So one application we use this idea for is this post-clustering differential gene expression analysis. This is a cartoon showing that in single cell data analysis, typically, once you have this count matrix, even though the orientation is different, here genes are put as rows, cells are put as columns, but it's the count matrix, I call data matrix. What we do is that since the cells don't have any labels, people rely on cell clustering just to cluster the columns here into cell clusters, hoping that they are cell types. But to confirm that, they perform some differential expression testing, hypothesis testing, to find the genes highly expressed in each cluster. And using those genes to annotate cell types, like each cluster will be given a cell type label based on the highly expressed genes to match the genes to knowledge to give a name. That's the standard practice. But from a statistical point of view, this approach used the same data twice, and it's called the double dipping problem. And I know several people here study this problem, including Daniela Witten, right? They have a recent paper here using the split count idea. And earlier, there was a paper by a Stanford group, which tried to use selective inference. So this problem will have this result that even if the two cell clusters are ambiguous, not well separated, because you use some genes, but because they must be divided by some genes, then the genes driven the clustering would naturally be found as differential expressed genes. So therefore, in this case, you cannot simply use the genes to say, oh, they are two cell types. So our approach to counter this problem is to use our synthetic null data, assuming that our synthetic null data comes from the null distribution where there's only one cell type. And what I see for our idea is actually, data-driven approach that mimics the negative control in real experiments. You have a negative data set in parallel to a real data set, you do the same computational pipeline, you compare the results in the end. That's one idea. And the second thing I can draw connection to classical statistics literature is the likelihood ratio test. We can think about the synthetic null data to be from the null hypothesis. We try to fit this distribution to our data to call that the null likelihood, right? And this is the unconstrained likelihood. We compare two likelihoods to draw a decision, I think we kind of implement the same idea in a numerical way. So in this way, we basically make the null hypothesis into data. And then people can look at the null data and decide whether it's something reasonable or not. I just think we make the null hypothesis more transparent. And the good thing about this is that in the middle, we can use any pipeline of user's choice. We don't have to constrain the clustering algorithm they use or the DE test they use. So basically, roughly speaking, for every gene, it will get two p-values, one from the real data set, one from the synthetic null data set, and we will compare the two p-values. That's the thing we do in the last step. So without talking about details here, I just want to say that for the last step, we do a contrast or competition-based FDR control instead of p-value-based FDR control. And this competition-based FDR control, the theory for it is from the knockoff literature. And also this idea has been previously used by proteomics people for controlling their false discoveries. So due to the time, I will quickly show some results about how this approach worked. So suppose that for every gene, we can have a DE score, which we define as negative log p-value from the real data. Similarly, we can define a DE score for the same gene from the synthetic null data. So every gene has a target DE score and a null DE score. What we define as a contrast score is their difference. So what I want to show here is if we have a set of known marker genes between two subtypes people know, and we have a set of housekeeping genes that are supposed to be highly expressed in every cell, and they cannot be cell type markers. For these two groups of genes, if we look at their target DE scores, just the real data analysis alone, they have some overlap. Some housekeeping genes may have very large DE score, and they are not the discoveries we want. But by doing our contrast, we can show that using the y-axis, the contrast score, the two sets are better separated. So the reason is for those housekeeping genes, even though they are unimodal, they have unimodal distributions, but they have kind of large variance. That's why they will be involved in dividing cells into clusters. Because they're involved, then they will be found as D genes. That's just the rationale or intuition. And these are two genes, one as a marker gene, one as a housekeeping gene. And what we do here is that we compare the genes' relative ranks in the result, like it's a ranked DE gene list, like top one is a top discovery, the bottom one is a bottom discovery. We compare the ranks. And here the ranks have a distribution because we consider different combinations of statistical tests used in the DE step and also different replicate data. So we're going to show some distribution. We can see that cluster DE can have much better separation of the marker gene in the top from the housekeeping gene rank in the bottom, while Surratt's rank distributions are less distinguishable. Furthermore, again, using those two sets of genes, we perform the so-called gene set enrichment analysis, which is a commonly used bioinformatic analysis to see where this set of genes is located in your top ranked gene list. And we see that marker genes are ranked more in the top in our result. Housekeeping genes are more ranked in the bottom in our result. In the popular Surratt result, their enrichment or location in the rank list are pretty similar. Second example, this example has a lot of cell clusters. This was a Nature paper, and interestingly, for Drosophila visual system in the brain, interestingly, the original authors defined more than 400 cell clusters. But for some of them, the authors couldn't give them different labels because they don't really look so different biologically. So we picked those examples to validate our approach. So these two clusters have the same label T1. These two have the same label TM1. These two have the same label TM3. So we just run our cluster DE on the two things here, and we didn't get any DE genes. But using Surratt, we can have hundreds to thousands of DE genes. And we use this plot to confirm that there are indeed no strong evidence to support their separation as distinct clusters. Furthermore, for two clusters with different labels, we also confirm that using the genes we select, we can have a much better classifier for distinguishing those two cell types than just using the Surratt genes. And of course, both are much better than using random genes for classification. And this heat map also shows that using our genes, the two cell types have much better visual differences. So all these results are in the preprint, and Koshing is here, so she is the co-first author for this paper. But we have done something more beyond this single cell analysis. That is, for spatial transcriptomics, it also has the same post-clustering DE analysis problem. This is from this paper's illustration. That is, when people get a spatial transcriptomics, they want to perform some sort of spatial clustering to define so-called spatial domains. Then to annotate each domain with a label, they do differential expression analysis. So similar idea, post-clustering DE. But the question is whether the two domains are really well separated or the separation is arbitrary. Okay, to answer this question, we implemented the same idea by generating synthetic null data in which when we fit this Gaussian process, we intentionally make the degree of freedom very low. So the surface is very flat. It's not like have sharp changes. That's what we want. So therefore, here, even though the gene has the same trend, for example, from low to high, but the change is more gradual. It's not sharp. You don't have boundaries. If this is something reasonable as a null, then we can show that this idea will give us much clearer marker genes for the spatial domains than if we just run Surratt. Because for Surratt, I can see even though the gene is indeed more highly expressed in the boundary, close to the boundary, but the change is not sharp. You have a gradual change, but the genes we pick have much sharper or much better indicators of the domains. That's just the ongoing or the new results we added. Furthermore, we think this model can be generalized to perturb-seq data, which is another type of single-cell technology where people add perturbations by the CRISPR gene editing technology to knock out certain genes. Therefore, in every cell, you would know which genes are targeted. You have targeted genes designed by experiment, and we can extend our model to allow for estimation of perturbation effect. And I think for this case, maybe some causal inference problems can be asked. And this is our preliminary results led by my student Qing Yang. That is, we have some promising research showing that our synthetic data can roughly mimic real data. And furthermore, if we want to go beyond one person, but to generate multiple individuals' single cell data, we can incorporate the genotype matrix of individuals. In this case, the individuals will enter the design matrix. Right here, I'm just showing 5 individuals and their SNPs, genotypes. We can copy that into our model. And we also have some promising results. We call this assay design for population. And the only competitor so far in the field is called SplatPop. Splatter is also a simulator, one of the first simulators. We showed that by considering gene-gene correlations, we are generating more realistic data. And this is led by my student Chris and also a new student Yihui. So for future work and several open questions, I think so far all our results didn't actually use uncertainty quantification yet. So even though we can theoretically do that, but we haven't used that. We are just using the point estimates. And model diagnostics, besides just checking this similarity between synthetic data and real data, can we have some more principled approaches? Not just the index I introduced, because the index relies on dimension reduction. So that's something I think is an open question. And something we must address is the computational efficiency. How can we make it faster? I think some simplification or approximation is needed to make the simulator fast. And finally, this is the thing I mentioned, the biological meaning of gene-gene correlations. What does it mean? Because you see in the UMAP plot, even though it's not accurate representation, but still we can see some cell types are more round and some are more narrow, indicating that the gene-gene correlation are kind of different. And what is that? What's the science behind it? So these are the questions I think are subject to further studies. Finally, I want to thank my group of people, especially Dong Yuan for leading the major efforts in the simulator and the resulting cluster DE, Ke-Xin, who is here, for contributing to cluster DE, and Christy, who has biological training, and she has helped us a lot on the biological data analysis. And what's very impressive is that Siqi is a visiting student. She just stayed with us for 1 year, and she contributed to the spatial transcriptomics part. Yeah, and of course, the Chris and Yihui for the population level single cell RNA. And also my former student Xinzhou, who is currently at Oregon State. Yeah, thank you very much.
Thank you for the brief talk. I have a question about your SC design probability of systems.
Yeah.
One thing I consider is different systems.
Yeah.
And other conditions.
Yeah.
Different needs, different settings.
Exactly. Yeah.
How do you
Very good question. So that's the good thing about the copula. So we can model every gene separately. So for each gene's distribution, it's conditional, it's using its own SNPs. And we just use the copula to link the marginal distributions together. Yeah. Yeah. Thank you. Yeah. Yeah, very good question. Yeah.
That's very cool. Yeah, I have a lot of questions.
Yeah, we can talk over dinner. We're going to dinner together.
Well, my first question is like, so the simulator does not typically not
Well,
usually like simulator will have smaller units compared to what.
definitely. Yeah, we also have the same thing. Yeah, because the real data may have some, you can call it outlier if it doesn't agree with your assumed distribution, right? These outlier values will definitely make your real data some, for example, if you look at here, right? So like these kind of things. Yeah.
Yes.
Yeah.
Also, like beyond outliers, like
Yeah.
there is unexplained variation.
Yeah.
Of the data by the model.
Yeah.
Then basically like the model still has smaller units. when you generate data.
Hmm.
It's also like smaller.
Actually, let me see, because in the current way of modeling, the variance parameter estimate of our marginal distribution, let's say, is just estimate from the data. So even though the model assumption might be wrong, but we're still trying to preserve the variance. Yeah, but I think what you're saying is maybe how do we decompose the variance? Decompose the total variance, whether you have some missing covariates. I think that's another problem. Yeah. But it's possible that we could have multiple models that fit well to the data. Then it became like a model selection problem, whether we should reduce the complexity or add more complexity. Yeah.
Another question, I was asking,
Yeah.
so like you mentioned different changes in expressions, like some, different.
Changing correlations. Yeah.
So how that was reflected in the resulting population?
A different gene correlations are different. I was saying different between cell types. Yeah, I was saying different between cell types. And that's a good question. So that's why when we fit the Gaussian copula for the discrete cell type case, it's done one per cell type. So within that cell type, we fit an m-dimensional distribution. So the Gaussian copula is also conditional on the cell type. Yeah.
But technically, the correlation standard depend on the cell.
That's what we are doing. Yeah, that's what we're doing. Yeah, but then you can see if the state covariate is not cell type, it's a pseudotype, right? Then we can decide whether we should fit just one Gaussian copula across all the pseudotype values, or we have to be conditional on a pseudotype value. Then the model will become very complicated. But rough way to do that is that you can potentially categorize or divide your pseudotype values into bins and fit one copula per bin. Yeah, that's such a rough way to achieve that. Yeah. Thank you. Yeah. Yes.
Curious about some of the details of time construct and all institutions.
Yeah.
So, specifically, like, so just reading out in the more complicated spatial.
Right. Yeah.
So are you taking observed data and in some way projecting it onto the model?
So for the single cell example I talk about, that one I talk about in detail, which is one we implemented, we just fit a multivariate negative binomial distribution to the data. Yeah, that's what we did. But I think for more complicated scenarios, if we have more than one covariate, some are continuous, I think some of the conditional randomization techniques might be useful to generate the null data. So you basically try to disrupt some relationships you are looking for. You make sure there are no such relationships. And then you just run the pipeline in parallel. I think the key thing I hope to achieve is that we can make sure this idea can be applied to however complicated the pipeline is. Yeah. Because essentially when we talk about the FDR control, we consider the whole pipeline, not just the final statistical test. Yeah. Yes.
Can I ask a question about mechanisms?
Sure.
That's what in reality is that
Yeah. Very small, yeah. Good point. So when you say recover, I don't think what we did can do any recovery. We were just estimating. So basically, if the sample correlation, the observed correlation is low, what we estimate will be low. We didn't do any enhancement. But if you want to generate, variants or alternatives to your data, you can arbitrarily set that correlation to be high and generate the corresponding data. You can do that. Yeah.
Also, the model starts
Yes, that's the key. We try to fit the model to real data and then generate data from the fitted model in a way to check whether the model is reasonable. Yeah, and once you think the model is reasonable, then you can modify its parameters to generate alternatives.
So fit the model.
Yeah,
So basically use a model to simulate.
yeah.
You haven't talked about.
Yeah.
Ever interpreted coefficients in the model.
Oh, yeah, actually, I think this is what I aim to see. This is what I aim to say. So I'm trying to interpret the point estimates without the uncertainty quantification here. Yeah, so this is like the correlation. This is like fitted spatial mean function for each gene. This is fitted mean function for each gene along the trajectory. Yeah. Yeah.
Yeah. You want to versus the slide.
Yes. Yeah.
Are there some applications that?
Yeah.
From that fascinating?
Yes, let me try to grab my another set of slides. I have that. I have talked about it in a different talk. Let me just see whether I can put. Okay. Yes, let me see. Is this one? Yes, let me just quickly show. And in that one, I talk about the Reed simulator. All right, so for this one, what I think as will be useful, let me just quickly go to the application, is that if we simulate single cell ataxic reads, we can change the read location. So basically, this slide is just showing that we can mimic real data. That's just it. But you can just generate alternatives. Where is it? Sorry. Oh, I'm not including here. Okay, my bad. Let me see. Okay, so basically, this is the one we can do. So we can mimic the real data, but in this generation, we specify where the peak locations are. So like here, we specify this is the peak, this is the peak. So using our defined peaks and non-peaks, then we can do some benchmarking. Yeah, and also we try to mimic the simulation or not simulation, sequencing error and the K-mer frequency in the real data. So just try to make sure the reads are like real data. And recently we got contacted by a user, which I think whom is from UBC, who tried to use the simulator to do the so-called demultiplexing, which means that you measure multiple individuals together and you try to separate their reads by individual because you don't know which cell is from which one. So you try to do that demultiplexing. And I think the simulator can also be used for benchmarking those. Yeah. Thank you.
There are no more questions. That's.
Thank you.